Let's see if can you see it in presenting mode? Yes?
Yes.
Okay. So thank you so much for this very nice presentation, Sarah, and thank you very much for this invitation to be here and to share a little bit of the work we do on grasspea in my lab. And I choose to tell you a little bit about my tendrils, like a tendril grasspea journey, because it had been somehow moving around. And maybe this can also inspire some of you, especially the youngest ones, to work more on this incredible plant. So how did I start working with grasspea? This is a picture of Europe. I hope you can see my, let me see if I can find, can you see my mouse?
Yes.
Maybe not. Yes? Well, I
Yes.
put it in a different color. Maybe it will be easier to see like this. Okay, so this is Portugal. And this is where I studied agronomy many, many years ago in Lisbon. And after I finished my agronomy degree, I decided to move to Spain, to Zaragoza, to this Mediterranean Agronomic Institute, where I did a master on plant breeding. And with that master, I had the opportunity to then do my project master in Cordoba, in IFAPA, where I started to work with legumes, in this case, faba beans, and on plant quantitative genetics. So later on, I moved to Wageningen in the Netherlands to do my PhD, and where I learned more about plant quantitative genetic, but at molecular level. At the time, working with wild barley. And later on, I moved to Italy for my first postdoc, where I went on studying molecular plant quantitative genetics, but with maize. And at that time, I decided that legumes were indeed my favorite ones. So I established a project between IAS in Cordoba and my present institute, ITQB Nova in Portugal. And I moved back from Italy to, in this case, Cordoba. And this is where I started to work with grasspea. So at the end, I finally move in to Portugal in 2007, and that's where I established my group that you see over here, the PlantX lab, or the Genetics and Genomics of Plant Complex Traits lab. We are a bunch of people. All of us, we work a little bit on grasspea, some more than the others, but we all have some work related to grasspea, and we are all very fan of this particular plant, but especially the grasspea PhD students, as Rita over here, she's working on grasspea disease resistance, and co-supervised not only by me but also by in a cooperation with the TSL in UK with Sophie and Camoon. Sara is a visiting PhD student from Spain, from the group of Diego Rubiales, and also co-supervised by me. Francisco is a PhD student together with Diego Rubiales also. So now he's in Spain. That's why we exchanged Sara by Francisco. That's why he's not on the picture. And finally, also Mathilde, that is also co-supervised by, in collaboration with Frank van Brozingen from Ghent University and the VIB, so also collaborating in grasspea, also with Belgium. And I have also to highlight the past PhD students, now doctors, grasspea doctors from our lab, Nuno, Margarida, David, and Letis. And some of the work I will tell you today will be also from their work. So this is a traditional field of grasspea in Portugal. We use the white flower varieties here. That's the ones that our farmers like. And as you know, all of us that are here today know about the diversity that this particular species has. We call it a diverse wonder, because indeed not only the flower color, but also the seed and many of the traits that this species has is very diverse within the species. So this is the type of seeds that the Portuguese farmers like, the big seed and light colors, but you are probably also used to the darker colors and smaller seeds. So what we saw when we start working on this plant was that as in many other legumes, there is a low research investment, but in this in particular, And this led to an irregular and low yield compared to other crop species that are interesting for farmers. And many of this irregular yield is also due to stress susceptibilities to different stresses. And on the other side, at least here in Portugal, there is an attractive outdated food product. So consumers are also, the ones that know the species that not so well known outside their production regions, they also are not very attracted for the type of food products that are developed right now, more traditional ones. So we thought in our group that we need to better align these market concerns, and so the concerns of not only of farmers but also of processors and consumers, with the breeding objectives in such a way that the new varieties that will be developed for this species will be more attractive. And for that, we need to develop efficient and low-cost selection tools. And this is where my group mainly work on the development of tools, molecular or other type of tools to help or to support the breeding of grasspea in more efficient ways. So I will tell a little bit about this tenderless path that my group has been following on grasspea research. And as I said, the first time I started working on grasspea was when I was in Cordoba, when I went back to the Iberian Peninsula, and in a collaboration with the RCSIC. And in here, we mainly focus on disease resistance of this particular species. So we studied the resistance to rust and powdery mildew on two particular Lathyrus species, one of them grasspea, and the other Lathyrus cicera, or chickling veg. So we studied not only rust, that was the first one that we started to study because it was the most easier way to do it. I have to say, when you are studying plant disease interactions, there are a few that are more easy to study than others. These are aerial part disease. And we studied rust on one side, but particularly looking at partial resistance to rust to be more durable. And we also studied resistance to powdery mildew, another aerial part disease. And we identified, we were only working at the time with a collection of Iberian origin of these two species, but we could still identify very interesting sources of resistance to these two diseases. So later on, we started to expand the germplasm collection that we were using on Lathyrus sativus, and we requested material from different parts of the world through germplasm banks, but we also collected more accessions from my own country in Portugal, in the three main regions that I will show you in a moment that still produces grasspea. And at the same time, we decided that we needed to invest also on the development of molecular tools because there was not a lot or almost none at the time. We needed to have them to do our research properly. So as I said, this is my country, Portugal. And so we went through the three main regions that are more mountains regions where this grasspea is still produced. So in Algarve, in the south, in the mountain near Lisbon, and also in the mountain near Tomar. And this, you could see that we went with our car all over, talking to everybody that was still producing grasspea and learning about their stories, about their concerns, and about their seeds that indeed everybody was willing to share with us. So at that time, as I said, we were thinking about also developing molecular tools, but also we started thinking about not only of diseases, but on other problems that were starting to be a concern for our grasspea farmers through, and that was drought at that time. So we also tried to develop some more physiological but easy to use tools to screen for drought tolerance on Lathyrus species. And that was the work of Susanna. She was a master's student at the time. And then on the work of Nuno, that was a PhD student, already finished, a lot of different molecular tools were tried and developed. Some of them were concerned the transferability of molecular markers from related legume species into grasspea. But also we developed a few transcriptomes of plant disease interactions like rust and Lathyrus, or Ascochyta lathyri and Lathyrus. And that also gave us not only transcriptomic information and expression, differential expression information, of the interaction, but also gave us a lot of molecular data that we could use to screen for diversity. So later on, we start thinking, okay, we visit all that farmers when we were looking for their seeds and their concerns. And the main production region in Portugal is Alveazer, that you see here, the design of the logo of the village, the city, and their farmers. So we decided that we wanted to start working more close by the farmers too. And so we were keen on doing participatory research, and the first place where we established this kind of interactions was then in Alveazer, and we worked mainly with one of our farmers that is still working with us right now, and then I will tell you that, okay, not only powdery mildew and rust were on our objective, but we start working with the concerns of the farmers at the time, and that was Fusarium wilt, quality, and so we moved for the field. So we left a little bit the lab for a while, not completely, and we start doing much more research on the field with the farmers, as you see over here, and in our participatory field trials in Alveazer, where we characterize the collection that we gather from all over the world under their conditions with the help of the farmers. But not only field trial was done at the time, so a lot of work was also developed on using linkage mapping approaches in Lathyrus, because then we had already molecular markers that we could use to build linkage maps. And this is mainly the work of led by our postdoc, Carmen Santos. And she focused mainly on Lathyrus cicera, chickling veg, but then also on the comparison between cicera and sativus, the two Lathyrus species that are more close by, and she learned a lot from this synteny analysis. So she developed the first genetic linkage map for Lathyrus cicera using the molecular markers that we had obtained from the transcriptomic studies. And then she used that to study the resistance to rust and the resistance to different powdery mildew, and to understand the genetic basis of this resistance. And she went further in more fundamental aspects. She also looked for the sequence of the MLO1 powdery mildew susceptibility gene into these two different species. And she characterized it. As I told you, the farmers were concerned about Fusarium wilt. So we had a PhD student at the time, Margarida Sampaio, and she developed all her research around the resistance to Fusarium wilt. She first looked for characterized the different, pathogens, isolates that we got from the field trials, and with the ones that our colleagues in Spain, the group of Diego, had on P. And she compared them, because since we know already for some time, P and grasspea are quite close phylogenetically. There is even now a line of thinking that will make a change in the Latin name of P. It will not be Pisum sativa anymore, but it can become Lathyrus oleraceus. So you can imagine how close these two species are, and they share the pathogens. So she compared the Fusarium oxysporum isolates from P and from grasspea, and then she characterized the disease reactions. So if there was susceptibility or some resistance in the big collection that we have of grasspea at the time. And finally, she developed the first genome-wide association study to study the genetic basis of this particular trait, that was the resistance or the susceptibility to Fusarium wilt in grasspea, and identified interesting candidate genes for it. Then we moved forward, and presently, I can say from 2022, we established something a little bit more structured, that is our grasspea Portuguese living lab, the GP Port. And this has been possible under the funding of a European project that is Divine Food, but I will tell you a bit in a moment. And also with a more interaction with different farmers, not only one of them, but several from Alveazer region. So this is the association of farmers, but also of small processors that we are working with. And not only that, but we are also doing some work now with citizen scientists. So, in the region where we are located in Lisbon, in Oeiras, nearby Lisbon. And we are using this not only to do a little bit of research with the citizen scientists, but also to disseminate this culture among the general public that do not know it so well in the places where this species is not cultivated in Portugal. So, and here we start looking also for the adaptation of this species to our conditions in more detail, but also we looked for more work on drought and flood, because this is becoming really a big problem in our with our farmers presently, and also on resistance to aphids. And for that we studied mainly, we applied genome-wide association studies to study the genetic basis of these traits. So, but I will first introduce to you the Portuguese grasspea living lab. And here is where we are in Lisboa, or near to Lisboa, in Oeiras, two cities that are nearby. And in more or less in the north central part of our country, there is Alveiazre. And Alveiazre is a city that is known in Portugal for the capital of grasspea, capital do Xíxaro. Xíxaro is grasspea in Portuguese. And this is their flag. Grasspea is the flag of this city. And there we established a very, very nice collaboration with a group of people that are interested in doing better with to conduct, or to say, to take grasspea to a higher level in Portugal. And these are different farmers. These are different processors that are over here, small processors doing different, very attractive and innovative food products. These are also the researchers that are here, like me and my colleague, or the municipality. So the local authorities are also involved on this, and also the technical school that is located in that region. So it has been a very interesting collaboration, and a lot of new ideas and new projects that become are grown from this network. So here you can see that we are also developing field trials with the farmers of the two guys over here, of this region in grasspea, in very, very different conditions in between the olive trees. So it really, we had to adapt our field trials to the growing conditions of traditional growing conditions of grasspea in that region. And this has been under the umbrella of the European project Living Food. And our citizen science project with grasspea here located in our institute, that has been funded also by an European project, Impetus, and where we do much more dissemination of the culture to the general public that do not know it. And so we went on also on the more fundamental research, and we start to do more GWAS analysis, so to identify the genetic basis of particular traits in the species. And that has been the work, for instance, of our PhD that already finished, David, that looked first, characterized the diversity of resistance to two different powdery mildew pathogens, the pea powdery mildew pathogen and the clover powdery mildew pathogen. Both of them are able to infect grasspea. And so, first of all, he characterized the collection we had from all over the world to resistance to these two different pathogens, and then identify the genes or the genetic basis of this resistance through genome-wide association studies. And then more recently, this is just from this month, the 5th of March, we published, or Rita, our PhD, one of our PhD students still under development, she published a very nice paper on more fundamental aspects of the interaction between grasspea and powdery mildew. So Rita is more interested on the resistance gene and the diversity of resistance genes across different legumes, but with a very special interest on grasspea and powdery mildew. So I invite you to see this very recent paper. And just not to forget that all of this work that we are doing on GWAS, so on genome-wide association studies, is now being much more facilitated after a reference genome of higher quality has been published with the group of Peter Emmerich, where we also collaborated. And indeed, this is now facilitating much better the annotation of the candidate genes that we managed to identify and to do all the process of genome-wide association studies in this species. As I told you, we also did a little bit of work on quality phenotyping and the development of tools for doing this phenotyping on quality. For instance, the work that we developed with the group of Rosário Bronze, here done by Andrea. And she developed HPLC-MSMS methods to quantify in a very precise way the beta and the alpha isomer of ODAP in grasspea. This is not one of the traits that we are more interested in, but when we need to have a very exact quantification of these two compounds, it is important that we have a very precise method, and she managed to optimize this method to these particular compounds. And then on the other side, we have the work of Elsa Mesha, also from the group of Rosário Bronze, and she's much more on the nutritional aspects of grasspea. And she compared grasspea with 4 other different cool season legume species for the protein content, but also the protein quality measured by the amino acid proportions and the protein digestibility. And she's doing more on this type of work, and more will come in the near future. And I told you also that we are more into drought and flood and the climate change resilience of this particular species. And this has been the work of Mathilde, our PhD project, where she characterized this big collection that I told you, not only for drought resilience, but also for tolerance to flood. And so we could somehow, how do you say, clear out if what were the accessions that could be resilient to drought and the ones that could be resilient to flood. But even then, we could also identify some accessions that were resilient to both stresses. And that is really, really interesting. Yeah, and I would not say typical from this species, but very interesting to find this kind of material. In the case of Lathyrus, that also finished already her PhD very recently, we characterize more all this collection on morphological and agronomical aspects, so of their adaptation to Mediterranean conditions. Since we had materials from all over the world, this was quite important for us to see what materials that could be sources of particular interested traits, how would they behave under our conditions. And that was most the work of Lathyrus, but more is coming in the near future. So stay tuned. And just to finish, I will invite you to visit our web page in ITQB Nova, the institute where we work. And here you can see all the news and all the projects and all the publications that are appearing from our group. And with this, I would like to say thank you for your attention. Thank you to all the funding institutions, and thank you to all our collaborators from all over the world. I already mentioned a few of them, others are here mentioned. And especially thank you to the farmers that are still growing grasspea and the small processors that do not mind to collaborate with us. And also to the citizen scientists that are also helping us a lot in this disseminating this crop. And just to finish, I would like to invite you to join us in the next International Legume Society conference that will take place next year in the beautiful Dubrovnik in Croatia. And that's it. Thank you so much.
Thank you very much, Carlota, for this inspiring presentation. I think it's great to learn about your research scientific pathways and to see really how you target your research on the need and interest of farmers. So thank you very much. For all the participants, if you have a question, please write it down in the chat, and we will come back to the question later. For now, we can move from Portugal to India. where Dr. Anjali Verma will present her work on Lathyrus. So she's a plant molecular biologist. She specialized in functional genomics and genetic engineering. Last year, after obtaining her PhD, she joined the Open Doors Fellowship Program, sponsored by the Fernand Bain Fund. Through this fellowship, she was hosted for 3 months here in Belgium at VIB PSB, where she continued her research work on grasspea. Anjali, I let you the stage to tell us more about your research.
Thank you, Sarah, for the introduction. So I'll just share my screen. Okay, so hello everyone. So today I will.
Anjali, sorry, we cannot see your screen for now, or at least I can't.
Okay. Yeah.
Yes.
So it's there, right? Okay. Okay. So hello everyone. So today I will talk about the potential of grasspea as a future crop. And okay, so I began working with grasspea during my PhD, where I joined a lab of Dr. Pramod, who was leading the Lathyrus project at what was formerly called as National Agri-Food Biotechnology Institute, which is located in Punjab in India. And with him and along with Dr. Ajay Kumar Pandey, I completed my PhD, which was specifically on grasspea. So before I talk about grasspea and its potential role in future, I would briefly discuss about some current global challenges. So this is the data which I have picked up from the recent Global Hunger Index report. So the first challenge is the hunger and undernourishment. So as you can see that this data pinpoints at the severity of hunger, especially in the South Asian countries, as indicated by red, orange, and yellow colors. And according to this data, there has been an improvement in reducing hunger in the past 2 decades. But this progress has somehow become stagnant from 2016 onwards. And not only this, the cases of undernourishment have also gone up, so much so that around 800 million people are undernourished as of now. And if I talk about my own country, India, there has been significant improvement in reducing the hunger rates in the past 2 decades, but still a lot needs to be done to reduce these values. So another global challenge that is often talked about is increasing population. So by 2050, it is estimated that the population will turn around to be 10 billion, and this will lead to an increase in food demand by up to 60%. Not only this, the agriculture sector also faces its own set of challenges on daily basis, which are further intensified under conditions of climate change, that ultimately results in reduction in both the quality and quantity of the agricultural produce. So what can be done to mitigate all these challenges? Well, I see it as that one approach that can be used is crop diversification. And it is reported that there are around 50,000 plant species which are suitable for human consumption, but only 300 reach the market. So we need to explore these underutilized species. We need to improve their agronomic traits and promote their popularization among the common masses. And most often, these underutilized species are climate smart in the sense that they are better adapted to tolerate the climate change. So here are examples of some underutilized species, and Lathyrus is one of them. It's about which I will be discussing more in detail. So Lathyrus, commonly we call it as grasspea, and in India we call it as kesari dal. So it's a cool season legume which is mainly cultivated for food, feed, and fodder. And it was introduced around 8000 BC in Bulgaria in Europe. And it is suggested it was the first pulse that was domesticated at that time. And from there, it was introduced to other parts of the world. In India, we have 6 major grasspea producing states, which used to grow grasspea extensively up to 1961. But then the ban was enforced, and now the government is trying to revive the cultivation of improved varieties in these areas. Now, why grasspea? Now, grasspea has a lot of benefits. It's a climate smart crop. It can better tolerate pests and diseases. It can tolerate the acidic soils, the drought soils, as well as flooded soils. And if we talk about the nutritional aspect, it stands second to soybean in terms of protein content, which is in the range of 28 to 35%. And also, it is rich in essential amino acids, that is lysine and threonine, which are usually lacking in the cereal crop. So, by complementing it with cereals, it can provide a well balanced diet. And not only this, it also has homoarginine, which is beneficial for cardiovascular health, and a compound ODAP, which has been patented for use in China and US to control bleeding during surgeries. So, what importance does it hold for India? So, more or less, it is similar. It acts as a protein source for majority of the vegetarian population in the country. And as it requires minimal inputs for its growth, it serves as a backup crop in the extreme weather conditions regions of the country where the other crops might fail to grow. And being a legume crop, it also enhances the soil fertility. And it also supports the marginal farmers of the country where it not only provides as an income source, but is also part of their local diets. So if it has so many benefits, then why is it neglected? The major problem is the presence of a neurotoxic compound beta or DAP, which is found in its seeds. And it has been reported that when people consumed consistently these seeds for 3 months or more than that, they were affected by paralysis of lower limbs, which we call as neurolathyrism. And so was the case in the farm animals. So how do we address this issue? Well, it can be addressed by removal or reduction of neurotoxin from grasspea, but how do we do that? So I will talk about various scientific advancements which have taken place in India with respect to reduction of ODAP in grasspea. So one being development of low ODAP varieties through breeding programs. So Indian research institutes like ICAR, various state agricultural universities have developed low ODAP varieties, and also in collaboration with ICATA, they have released the low ODAP varieties which are suitable for the areas which I have already discussed previously. And one of the success story is the revival of grasspea cultivation in one of the Indian states, that is Bihar, where the government has initiated the program of capacity building of farmers to promote the cultivation of the low ODAP varieties, where it not only serves as their income source, but also as part of their protein source in the diet, and also provides feed block for feeding the farm cattle. Well, besides the traditional breeding approaches, molecular approaches have also been explored for lowering ODAP in this crop. So during my PhD, we were trying to characterize the enzymes, which are the genes which are involved in the beta ODAP synthesis in this plant. And we wanted to target them by CRISPR-Cas approach to produce lines with low or no beta ODAP contained in it. So this was the major objective of the study which I had undertaken during my PhD. So I'll just briefly explain the pathway. So there's a core pathway that gives rise to diaminopropanic acid. Some of the steps are still not known. And there's a plant secondary metabolite oxalate that gives rise to oxalate coenzyme A. In presence of enzyme, oxalate coenzyme is synthetase. Then both these compounds, they combine in presence of beta ODAP synthase to form ODAP. We were intrigued by the entry of this plant secondary metabolite oxalate in this ODAP pathway. Hence, we decided to characterize this particular enzyme, OCS. But when we started our work, we did not have any tools to perform gene characterization, as there were no stable transformation protocols to do that. So, do we have tools to alter the genome? Now we do, and that is the hairy root system. So, using this system, which we have developed in our lab, we have performed genome editing of this OCS gene using single guide RNA approach in 15 independent hairy root lines. And we have shown the editing percentage to be around 73%. Also, we have used the double guide RNA approach where we were able to delete a large chunk of gene in using this approach. Finally, these transgenic hairy root lines were then analyzed, where we noticed that on gene knockout of this OCS, excess of oxalate accumulated in these transgenic hairy root lines. So this data, which we have recently published last year, can be accessed for more information. Well, the story is still not complete because the system can be used to explore other potential targets so as to have ODAP-free plan for future. And another significant achievement was the release of reference genome of Fusa 24, which is a popular Indian cultivar here, and this serves as a strong foundation for carrying out the crop improvement programs in future. And if I talk about my experience with grasspea beyond borders, so as an open doors fellow, I had the opportunity to explore grasspea beyond India as well. But before I talk about that, there was one more aspect of my PhD study where we were trying to understand the role of ODAP with stress tolerance in this crop. And the findings which we have published in 2022, the objective was to integrate these findings with the stress resistance project that is being currently headed by Professor Frank at VIP. So by integrating the findings, the objective was to gain more insights into how ODAP accumulation occurs in this plant and how it is involved in providing stress tolerance to this plant. So further, as a VIB IPBO fellow, I also got the opportunity to visit Dr. Carlota's lab at ITQB. And also along with her, I visited the grasspea field trials in the farmer's field, where I was amazed to see that how, as a scientist, she is addressing the real world challenges which are being faced by the grasspea farmers. And not only this, it was my first time to taste some of the local delicacies which are made of grasspea, which were indeed great. And as a VIP IPBO fellow, I also got the opportunity to attend the first International Lathyrus Day in the beautiful city of Kent, where I had the opportunity to present my work and network with the global grasspea community. So in the end, I would like to acknowledge my PhD supervisors, Dr. Pramod and Dr. Ajay Kumar Pandey, and my lab members. And with this, I also would like to acknowledge IPBO team, the Open Doors Fellowship Program, the Finale Bean Fund for providing me this opportunity, and also the OSS group for hosting me in Belgium, which is led by Professor Frank, and also by PlantX Lab for hosting me during my stay in Portugal. Thank you.
Thank you very much, Anjali, for another inspiring presentation. It's great to see your line of research. We can now move to the question and answer. We have already collected some in the chat. If you have more questions, feel free to keep posting them here in the chat, or also raise your hand, and we will give you the words. So let's start with the question posted in the chat. Mary Franklin is asking to Carlota what so given that grasspea is considered a drought and flood tolerance crop, what is not found in the from the farmers you work with? Are there Portuguese varieties less tolerant than those used as insurance crop elsewhere? And the question is particularly interested in the contrast between the reputation of grasspea and what is lacking from the farmer's point of view.
Okay, thank you, Sarah, for this, and thank you, Mary, for the question. So, what I think is exactly like that. We had the opportunity to compare our traditional grasspea varieties from Portugal with varieties from all over the world, and also from not only from the Mediterranean area, but also from Asia and different countries. And what we realized was indeed our Mediterranean varieties with big seeds and light colors were less resistant to drought and to flood. And but it's a question of how much drought and how much flood we are getting right now, because before our farmers were really happy with these varieties, but presently we are getting a lot of flood events, so a lot of rain in very short time during sowing times, or just a little bit after immersions, and the plants that we have in the farmers' fields will not survive this flood. And on the other hand, we are getting less water all over the cycle of the plant, especially at the end so at the flowering and maturation of the seeds during spring. And so final drought is becoming a real problem in our conditions, in our Mediterranean conditions. And what the farmers are seeing is that their varieties, their traditional varieties, that they were very happy with them, and they select them for many, many years throughout across generations, are not so well adapted anymore to these changings that we are suffering right now. I don't know if it's climate change, but probably it is. Yeah, I think Mediterranean climate, it's always very unstable. That is typical, but it's becoming even more unstable than before. Yeah.
Okay, thanks.
That's really interesting. Thank you.
Thanks. So yes, speaking of drought, the next question is from Maruti Gouda. So the question is, what are the secrets of grasspea in respect of drought tolerance? Carlota, could you tell us more about research progress on drought-resistant characteristic of grasspea?
Okay, so for this, I will direct you to the paper of Matilde Sanchez. She's the PhD student that is together with Frank van Brozgen. And she really took a very detailed physiological analysis of the response to drought and flood of this huge collection of accessions from all over the world. And she measured more than 20 different physiological traits during these two treatments, water treatments. So there you can understand better what were the mechanisms, but above all, it has, if I remember well, it has more to do with the root to shoot ratio of the plant that is indeed providing better survival rates for the plant under this extreme conditions, and also to the water use efficiency. So the plants are more efficient to use water. But if for more details, I will ask you to see the Matilde paper, and it's open access, so you will find it easily. I can even put it in the chat, the DOI, okay, to get it.
Yeah, I think it has been posted by the participant. Thank you very much for your answer. Now, Renella Bemaro is interested in grasspea for cultivation and is wondering where the seeds can be found in Madagascar. Maybe you both have suggestions in this regards or in how to find grasspea seeds.
Well, I would say that probably you could request some seeds from germplasm banks, international germplasm banks, to test them in Madagascar conditions and to see what would be the ecotypes that are better adapted to your conditions, because they could be the darker, smaller seeds ecotypes, or the whiter and larger seed size ecotypes from the Mediterranean. And after that, maybe you will find a farmer that is still producing this, or some cultivated, some commercialized varieties in these particular regions of the world that will direct you better. So I would start by the germplasm banks to test the species under your conditions, because they only have a small amount of seeds, but you will have them to test. So that would be nice.
Okay, thanks.
I don't know if Anjali has another option for this, for the seed search.
So, well, yes, for the seed search, definitely we do have germplasm here established in a country where definitely people can request. In fact, the leading institutes, ICR and other NBPGR, which are there working on them, and also the centers of the Patto in India. So definitely these seeds can be provided. And also the institute from where I have done my PhD, we do have some seeds to share.
Perfect, thank you. Jamlik Mwanji is asking regarding intercrop. How does grasspea intercrop with other crops, especially in maize? And what is its contribution to soil microbial structures? So I think it's interesting to take this question and have it answered from both the prospect of Europe, let's say Portugal, and the prospect of India. So regarding grasspea and intercrop.
Okay, I can say that we made some testing intercropping grasspea with other cereals, but not maize, because maize in Portugal is a spring-summer crop, and grasspea for us is an autumn sowing crop. So they have different cycles in our conditions. But we tried out with different ancient wheats and spelt and rivet wheat and einkorn. I have to say that this is a mixed feeling results, because the farmers were not so happy with the results. But not because of the production by itself, but it became very complicated for them to harvest it and to separate the seeds of the cereal and the grasspea. They were not used to this kind of cultivation anymore, so they do sole crop grasspea. And so they decided that intercrop was not for them. So we could not follow this testing with our farmers. So I have not much more information about it.
Okay, interesting to know. Anjali, do you perhaps have information regarding intercropping in India?
Okay, well, like I mentioned, the cultivation has been banned since a very long time. And in 2018, the cultivation was introduced of the improved varieties. So, yes, in some regions, in the rice fallows, they might have been cultivating them. But as such, we do not have much data to talk about the intercropping system because of the extensive ban that has been imposed on grass species.
Okay, thanks. So maybe something that can be explored in the future. Cathy Martin is asking to Anjali, what is the phenotype of KOOCS root, and are they beta ODAP free?
So the knockout effect of OCS in these transgenic hairy root lines was we noticed there was excessive oxalate accumulation. But when we measured the ODAP levels, we did not see any reduction happening to that expected level. Now, there could be certain limitations with regard to the hairy root system, which we might have encountered. conclusively we cannot say until we test this in the whole plant system. So we in parallel that work is still going on where we can convincingly say what actually happened in the whole plant system.
Okay, thanks. Then let's go to the next question. How many varieties are released in Portugal regarding abiotic stress?
Well, I can say that we have no commercial varieties in Portugal of grasspea. So the only varieties we have are the traditional produced by the farmers. This is the Portuguese reality. We have chickling veg varieties commercially available, so registered in Portugal. So 3 different ones, if I'm not mistaken, but more for animal feed. But in Spain, there are several, so our neighbor country, there are several grasspea varieties registered. Yeah, but no, we are just starting. Okay, we have developed the methods to select the material, we characterize the collections, and we are now developing pre-breeding material. So we're starting process with interesting sessions with resilient to abiotic stresses, but we are not at that step yet.
Okay, thanks. Andalo Getnet is writing so that in their area there is the belief that the straw of grass pea can cause diseases that damages the legs of human being. And is asking if there is any scientific explanations in this regards.
Okay. So, well, there is no clear-cut explanation as to how it actually happens, but there are reports which suggest that it competes to bind with the AMPA receptors in the neuronal cells, and which affects the homeostasis of the cell, and that leads to some disruption with the calcium signaling pathway that ultimately affects the extracellular matrix, and hence then there are some locomotory problems which are caused in the human. So more evidence and more research is needed to convincingly say that, but yes, in preliminary results in the cell lines, this is what there is data which is out there.
Okay, thanks. But I guess this is also just the case when the diet is mostly based on grass pea, right? To develop this disease.
Yes.
Yes, you have. I think it's important to say that you have to eat it as a sole
Yes.
crop, as a sole food for like 3 months to get very
Yes.
sick. But also other legumes have this problem. So I think I don't advise anybody to eat just one particular legume in during 3 months. They will be
Yes.
sick for sure.
That's a good
Yes.
one.
Yes, and not to mention all this data is reported based on the fact when there were famine. So when people had only this source to feed on, so
Exactly.
such cases were reported during that time. But in general, I don't think so. This is happening in the present time anymore.
Okay, perfect. Thank you very much. Okay, so last question from our colleagues at PSB to Carlota. Have you identified a gene or a QTL for powdery mildew from grass pea? And if yes, have you transformed it to other crop to see its effect since the powdery mildew affects many crops?
Okay. Yes, we have clarified the genetic basis of 2 different powdery mildews in grass pea. So identify the genes or the QTLs that control powdery mildew resistance, in this particular case, partial resistance. So as a quantitative trait, it was based on many genes with small effects, or many QTLs with small effects. And what we can say, to make a long story short, no, we have not transformed it into other crops. But what we can say is that, for instance, the genetic basis of resistance to powdery mildew, and clover powdery mildew in grasspea, they are different. So they are controlled by very different QTLs. And both of them are complex, so many genes influencing the resistance with the small effects. So the idea is that transformation, to see probably a clear effect, you should not transform just one, but several genes or QTLs into another crop. And it's always the same problem. So you have a very complex genetic basis on these important traits, but then it's not a major gene. So it's not easy. Well, transformation is never easy, but it's not easy that you will find an effect if you just have a small effect on your own crop for a single gene. But we never did it, indeed. But maybe also because of that, it is quite complex, the basis.
Okay, perfect. Thank you so much. And I think we are approaching the end of this online cafe. So I would firstly like to thank our speaker, Carlota and Anjali. Thank you so much for taking part in this online session. And then all of the participants for making it so interactive. And I hope to see you at the next online cafe. If you have any question, you can contact us or our speaker. There are emails in the chat. Thank you very much.
Thank you. Bye-bye.
Bye.
Thank you. Bye-bye.